mechanisms of enzyme action. And next, in order to explain, in order to explain the mechanism of enzyme action, as a German chemist Emil Fischer proposed to can key model. Uh, according to this model, according to this model, both enzyme and substrate possesses specific shapes that fit exactly into one another. This model explains enzyme specificity. And in 1958, an American biologist, Daniel Coastland, suggested, suggested a modification to lock and key model and produced induction slash fit model. According to this model, active site is not a rigid structure, rather it is molded into required shape to perform its function. Included uh, induced fit model is more acceptable than lock and key model of enzyme actions. His upper part is substrate, his lower part is enzyme active site. And when it's attached, he made active site change its shape and enzyme substrate complex is short name when he attached to both sides. Enzyme and substrate and is active site and his full name is enzyme substrate complex and your fit model of enzyme action. And specificity of enzymes, there are over 2000 known enzymes, each of which is involved in one specific chemical reactions. Enzymes are also substrate specific. The enzyme protease which breaks peptide bonds in proteins will not work on starch, which is broken down by an enzyme amylases. Similarly, lipase enzyme acts only on lipids and digests them into the fatty acids and glycerol. Similarly, lipase enzymes acts only on lipids and digest them into fatty acids and glycerol. Specificity of different enzymes is determined by the shapes of their active sites. Active sites possess specific geometric shapes that fit with specific substrates. And how the geometric shape of active life of enzyme determines its specificity for substrate. Point out which substrate can exactly fit in the active site. Substrate 1, substrate 2, and substrate 3 is different ironics and is practical work. Perform an experiment to show the working of an enzyme in intro. Enzymes can catalyze in vivo and in vitro reactions. Vivo and in vitro reactions. We can design an experiment to observe the in vitro enzyme activity. For this purpose, we will select meat proteins as substrate and pepsin as the protein digesting enzyme. That is problem. Can pepsin digest the proteins present in meat and is apparatus required? Meat, test tube, test tube, pepsin solution, HCl, and burette reagent. And background information, in vitro means outside living body in artificial environment, while in vivo means inside living body. And animal flesh meat contains lot of proteins. And the procedure, Take take a small piece of meat into test tubes and pour 15 ml of pepsin in one of them and pour 15 ml water in the second tube for comparison. It's just comparison. Number 2 point, add 10 drops of HCl in both test tubes and place them at 37 degrees C in incubator. Observation, observe the piece of meat after 4 hours. Perform the Biuret test to confirm the presence of proteins in both tubes. Go to chapter 8 and season 8.2 for the procedure of Biuret test and its results. The Biuret test gives negative results in the tube in which the pepsin was added. It confirms that no proteins are present in this test tube and all have been digested by the enzyme pepsin. And its evolution. What effect did pH have on pepsin activity? Number 2. What is the optimum pH for pepsin? It is the pepsin evolution results observation procedure and is practical working of all information. So my friends, how is my topic today about, which I teach you about what is the mechanisms of enzyme action. I hope you like and take good knowledge. So if you are like, so also please subscribe my channel and press the bell icon, because when you do press the bell icon, then you receive my all best new contents for your YouTube page, and you will also see my best wonderful just topics in this your YouTube page, and you will also feel happy when you see my all knowledgeful information topics when you see in your YouTube page. So now you give me permission. I am go to my next new topics. Take care, Allah Hafiz. Me and you, we will meet our next new best topics, and then we I made best video for you, and you will also receive good knowledge, inshallah. Ta'ala, I hope. So now I am good. Take care, Allah Hafiz.